like it is basically joint families are good stating, but sometimes small children also may get less attention due to joint family from their mother's perspective. So that.
Good afternoon, everyone. So thank you for giving me the chance to present here, and thank you for. So my topic is about comprehensive quality and control analysis of biobank samples, assessing nucleic acid integrity and purity. So our just I would like to conclude that before dive into my work, this hospital is about congenital dedicated to congenital heart defects. So congenital heart defects is abnormality which is present since birth. So CHD includes simple septal defects to complex single ventricle morphology, where in the figure you can see septal defects like atrial septal defect, ventricular septal defect. In conotruncal defects, stop PG are there, and left side obstructive lesions like HLHS, bioaortic wall, COA. So CLD is one of the leading cause of childhood mortality and morbidity. So in India, birth prevalence is 9 per thousand live births, so resulting in more than 2,40,000 children born with CLD each year. So some of the critical cases like one fifth of needs intervention is during their first year of life. So uh, in India, pediatric cardiac care faces more challenges like financial limitations, where the patient cannot afford costly treatment, then the general lack of awareness, unequal distribution of specialty centers providing quality care. So, to address this, the National Public Health Problem of CHD in India, Sri Satyasai Sanjeevani Hospital has the largest chain of pediatric cardiac care hospitals. So, it is located in four centers. We have the four centers from the past 12 years, like one in Maharashtra, Haryana, Chhattisgarh, and Telangana. So about 35,000 surgeries and care interventions have been performed, and about 3 lakh outpatients have been managed. So with this, at Sanjeevani hospitals, all services are totally free of cost. So considering large clinical volume, we have diverse spectrum of CHD cases, and on an average, 10 to 12 surgeries and cath interventions performed per day per center. So there is also a voluntary participation of patient families to understand the cause of the disease, and they also provide the samples. So Sri Satyasai Sanjeevani Research Foundation working closely with Sanjeevani hospitals. So for that, we have established three repositories at our three centers, like in Palwal in 2018, in Kharghar 2023, and in Raipur 2024. So this all entire work happens under the guidance of National Ethical Guidelines for Biomedical and Health Research involving human participants 2017, which is laid down by ICMR. And also we are working in the boundaries of research advisory committees, institutional ethics committees, biobank regulatory committee, and biosafety. So we have this sample size at our 3 centers. So in Kharghar, where I'm working as a biobank coordinator, so we have 10,552 samples, and for all centers we have 46,804. So this is the splits from our Kharghar biobank, where we have patient sample more than 1,800, and for blood and serum from the trials, which is more than 4,000, and tissue samples we are getting from only patients on the discretion of our surgeon. So it is near to 1,500. Twin samples where one or both the twins are affected, those samples we have 10. Siblings, available siblings, we have 22 samples. Sporadic cases, 1792. And syndromic cases, we have 12. And we have more family cases, but familiar cases, but in that, for the entire family, the count is 1. So another graph shows about cyanotic and acyanotic, where CHG is broadly classified into cyanotic and acyanotic, where it shows acyanotic is a more predominant in the patient. So the trio sibling and twin samples provide an added advantage for defining the cause of CHD in future. So the next graph shows subgroups of CHD classification, where we in our data we have CHD cases more than the other subgroups, where it is 35.16%, following by top, which is 21.74, and so on for the other groups. So we have more surgery cases than the CAT. So in India, VSD is the commonest of acyanotic CHD. Moreover, top is the leading in cyanotic CHD. So this is our biobank workflow. When the patient visits to the hospital, registration first registration is done, and then clinical records are maintained. So as a gold standard for CHD diagnosis, 2D echo is done, and with this confirmation for the CHD, sample collection is done for the lab assessment. So whatever the leftover sample is there, it comes to our research lab, where we call to the patient parents and we do consent. So sample and the data collection is also done from the parents, and unique identifier like code and labeling is done to the all the samples. Then sample processing is done, and we do multiple aliquots for those samples, and then these samples stored to -80 for further research and for quality control. So some subset of these samples we use for the quality control, and we also revisit our sample and the data collection to improve if it is required. So as a biobank coordinator, I also look into the quality control measures, where we do a pre-analytical quality control. So we also ensure that ICD forms should be completed. Then cold chain temperature monitoring till sample reaches to the lab. Then generation of code and cross checking is also done. Then storage QC, like we also have freezer temperature log. Then sample collection and storage QC is also done, where we ensure integrity of the nucleic acid and metadata accuracy. We're ensuring correct sample annotation and donor history, and the details are also made sure. So as a normal routine process, we also do the quality control for DNA and RNA. But for this particular study, we have sent some subset of samples to the Strand Life Sciences from Bangalore for further validation. So this is the result, where it shows the quality control of DNA. So 97 blood samples plus 31 tissue samples are isolated for that to calculate the D value. So 28 samples falls in the range of 5 to 7.5, and 100 samples falls in between 7.5 to 10. So all samples pass QC, and further it is processed for library preparation for whole genome sequencing. So next is the result for the RNA. So same 97 blood sample plus 31 tissue samples are also isolated for the RNA, where 68 samples fall in the range of 5 to 7.5, 45 samples falls in the range of 7.5 to 10. So 92.96 samples pass RNA QC and further process for library preparation and RNA sequencing. So to conclude that 100% of DNA samples and 92.96% of RNA samples pass QC, which reflects high quality collection process and storage of samples in our biobank. And the limitation to the study observed that 8% of failure rate for surgical tissues. So therefore, there is need for potential. So the potential reason for RNA degradation need to define for more improvement in the yield of and quality of RNA. So I would like to thank to our chairman sir, our director sir, and our most special thanks to the patient and their families, and our collaborators and scientific advisor from ACTRA, Dr. Sanjay Gupta, and our group leader, Dr. Radhakrishnan, and also to the thanks to the organizers who given me chance. Thank you. that is called RNA. So RNA should be handled uh, as RNA. We know that it degrades quickly. Uh, so we have to maintain it as possible as we have to maintain it temperature, and we can store those samples properly in our biobank. Yes. Actually, when we have this list of daily list of surgery and CAT. So for those only parents, for those patients parents, we call daily basis. We call them, and then only we collect samples from them. So just we have to call them, and we have to tell them about how severe this disease is and what are the factors responsible. So uh, hmm. Actually, we get very nice this thing percentage. Means 100% is only 1% we can leave. Otherwise, we take. Okay.
All participants are winners, okay? Forget about the prize.
So, good afternoon to all. My name is Anshul from AIMS Biotechnology Department, AIMS, New Delhi. Today I'm here for my study entitled MYH9 mediated cell cytoskeletal anomalies lead to thrombocytopenic features during the dengue virus pathogenesis. So, dengue virus is an RNA virus and it's a causative agent of dengue disease, which is classified as dengue fever, dengue hemorrhagic fever, and dengue shock syndrome. The key clinical feature associated with this condition is the thrombocytopenia, which is simply defined as a low platelet count. And we can clearly see the decrease in the platelet count with the increase in the platelet, with the increase in the disease severity. So the literature has suggested that the dengue-associated thrombocytopenia could be arising due to the dysregulation of the megakaryopoietic process. But the exact cause behind the molecular cause behind this phenomenon is still not elucidated fully. So in my study, I have focused on elucidating the molecular events that could arise the thrombocytopenic features during the dengue virus infection. So for this, firstly, I have initiated with the gene expression profiling of the megakaryopoietic genes, and from our data, we found that the MYH9 is significantly downregulated during the dengue virus infection. So from this, the MYH9 suggested as a good candidate to proceed with. After that, we have further validated it with the QRT-PCR and the Western blotting, which suggests the decrease in the expression of of this gene at the RNA and protein level. The protein expression is getting affected. This might be affecting the function of the MYH9 genes, which is a cytoskeletal. So the dengue infection must have the effect over the cytoskeletal integrity. So to check this, we have performed the immunofluorescence assay, and our assay suggests the same. In the control cells, without dengue infection, the cell integrity and cell shape is maintained. But during the dengue infection, the cytoskeletal integrity has disintegrated, and we can see the more condensed and disorganized actin fibers. So from this, we concluded that the dengue virus leads to the changes in the cell cytoskeletal integrity during the infection. After that, we search for the literature and to emphasize the role of this gene as an important candidate. So we found that the MYH9 is critical for the platelet biogenesis, and particularly, especially, this platelets exclusively producing this MYH9 for the cytoskeletal maintenance. So we hypothesize that if any change in the expression of this gene must have affected the process of platelet generation. After that, apart from this MYH9 is so crucial for the is responsible for the bleeding manifestation disorders. Any mutation in this gene has leads to the bleeding disorders, and it has its own class of clinical disorders, which is termed as the MYH9RD. After that, the question arises how this important gene is dysregulated during the dengue pathogenesis. So to answer this, we search for the transcription factors that can regulate the expression of this gene during the dengue infection. So we searched the in silico analysis, and we found that the RUNX1 is a good transcription factor that possesses the three potential binding site in the promoter region of this gene, and it has a potential to regulate the expression of this gene during the dengue infection. After that, to gain some confidence in the physiological relevance of this gene expression, we perform the gene expression analysis from the patient samples, and this data has interestingly suggests that the this MYH9 gene has a potential to be served as a biomarker. That we can see the expression of this MYH9 gene is decreasing with increase in the disease severity, but we have to conclude this with the broad clinical samples. After that, because we investigated the interaction of RUNX1 with the MYH9 promoter. So for this, we first clone the MYH9 promoter and then performing the luciferase assay and the chip assay to emphasize the role of dengue infection in affecting the interaction of this RUNX1 transcription factor with the MYH9 promoter. And our chip data and luciferase data suggests that during the dengue infection, this interaction getting hampered, and this due to the decrease in the interaction of RUNX1 transcription factor with the MYH9 promoter, it leads to the decrease in the promoter activity of this MYH9 gene. After that, this RUNX1 is regulating the expression of MYH9 gene. So we knock down this RUNX1 and then checking the effect of the knocking down this RUNX1 over the expression of this MYH9. And we found that if we knock down this RUNX1 transcription factor, the MYH9 expression is getting decreased concomitantly. After that, we also check the what is the replication status of the virus if we knock down the MYH9 expression. So we found that if we downregulate the MYH9 expression, it supports the viral replication. After that, since our study till this that our studies performed on the HUS7 cells, which do not originate the platelets, so we move towards the better cell line model to investigate the effect of this axis over the platelet biogenesis. So we chose the K562 cells for our study, and in these cells can originate the platelets under the PMA induction, and we further established this model in our cell line in our experimental condition, and we checked the PMA induction with the ginside staining and the PI staining. PI staining suggests the increase in the ploidy of the ploidy of the cells, and the ginside staining suggests the increase in the size of the cell. After that, we, in collaboration with the Dr. Shankar Bhattacharya, THSCI Faridabad, we performed the RNA-seq analysis, and our RNA-seq analysis suggests the differential expression of the genes which participated in the megakaryopoietic process. And interestingly, this data also suggested that not only affecting the MYH9 expression, the virus having a multi-channel effect over the megakaryopoietic process, where it affects the gene expression of the proliferative gene, the proliferative marker that associated with the megakaryopoietic biogenesis, and it increased the expression of the apoptotic marker, that is the CYCSD. So from this, we conclude that the virus has a multi-channel effect over the platelet biogenesis process, and it can affect the platelet generation via multi-channel effect. After that, we the next one transcription factor MYH9 promoter axis. We have established this axis in K562 cells, and we first optimize the transfection. Since the K562 are the suspension cells, so they are difficult to transfect. So for this, we first optimize the transfection, then perform the luciferase assay and the chip analysis. And our analysis suggests the same, that this working axis is working also in the K562 model. So after that, these are the key observations from my study, that the megakaryopoietic genes are differentially expressed during the dengue infection, and the MYH9 is protein which is significantly downregulated during the dengue infection in both the HUS7 cell lines and in the K562 cells. And this protein play a crucial role in the platelets maturation and the release. The third is the expression of MYH9 and RUNX1 is progressively decreased with the increased disease severity. Thus, could be served as a potential biomarker for the dengue disease severity. After that, decreased MYH9 expression leads to the cytoskeletal anomalies in dengue infected cells. MYH9 promoter activities under the control of RUNX1 transcription factor, and the dengue infection disrupts the RUNX1 binding to the MYH9 promoter. Lastly, the dengue virus infection leads to the reduction of the platelet production and upregulates the platelet destruction marker, that is the CYCSD. This is my working model where I have suggested. Lastly, I want to thank to my PI and the lab and the funding.
One question. Is there any role of this gene to make any capsid of this virus?
No, not exactly, because till date there's no study which suggests that we do not suggest that the host factors are directly responsible for the for maintaining or formation of the capsid.
Like the condition. You suggest for this.
What kind of a food? So
Something else.
yes, but
Because
yes, so like what kind of food? Like the diet, I do not know the preferentially what kind of diet the dengue patient should have, but yes, sir. No, the exactly the food which increase the cell integrity or like that have no effect on the medicine. Yes, sir, during the viral infection, like particularly the dengue infection or RNA virus, they do not known that the virus cause the direct mutation in the genome. So, what kind of mechanism they apply is that they regulate the gene regulatory mechanisms like the polyadenylation, like the splicing analysis, like the post transcriptional modification. So they prefer to modify the post transcriptional modification or the gene expression, but there's no report that directly caused the mutation in the genome. Yes, sir, these are the literature which suggests that what is the importance of this MYH19 for in the sample, so that the MYH19 emphasize that the MYH9 could be a potential marker because this gene is solely responsible for the maintenance of the cell cycle. Thank
No, yeah, Dr. Mangla Gauri. Mangla Gauri.
Yes, I'm online. May I present? Am I audible?
Yes, yes.
Yeah. Please let me know once my screen is visible.
Hold for a moment.
My screen is visible?
आप कुछ open कीजिए मैं बता अपने your presentation kindly upload your presentation now.
Yes, yes.
There is a presentation tab below the app.
Yes, yes, I have done it.
But your presentation is not uploaded yet.
I think it's processing.
Okay.
Is it visible? My slides? Hello. Am I audible? Whether my slides are visible? Yes. Good afternoon, everybody, respected dignitaries. My topic is regarding the ethical complexities in the usage of the residual. Am I audible? If you can confirm. Regarding the ethical complexities in the usage of the residual. Am I audible? If you can confirm regarding the ethical complexities in the usage of the residual. Am I audible? You can confirm regarding the ethical complexities in the usage of the residual. Am I audible? You can confirm. So my topic is on the ethical complexities in usage of residual blood samples for the research purpose. Coming to the introduction, it is very common there is a collection and storage of human blood biological materials have become an integral part of the healthcare system. So these biological samples are collected and stored for various purposes like the diagnosis, treatment, and even for the donation. And among these, the blood is the most commonly available biological sample. And these blood banks are the very convenient place for the taking up the research, as there is a lot amount of documents are reduced and blood samples are available. And also, this is an opportunity for the researchers to carry out the research to contribute for the advancement of the medical sciences. Along with this, there comes a lot of technical and the legal and ethical issues which will be taking care handling these biological samples. So the ethical issues, the next slide. I'm controlling. Yes. Okay. So there are some ethical issues which come up during the residual blood samples handling. Those are the informed consent, storage, confidentiality, individual autonomy, export ownership and commercialization of the stored blood samples, and also the trust in and the credibility of the researcher and also the lab which they are handling these samples. So this current study was taken up to emphasize the attitude of the donors and the patients in use of their residual samples and the stored samples for the research purpose. So, coming to the aims and objective of my study, here we are assessing the attitude of the patients and the blood donors regarding the use of residual blood samples or the stored blood samples for the research purpose. And we are comparing the attitude scores between the donors and the patients. Coming to the materials and methods, it is a cross-sectional study conducted in the private hospital and the general population. The questionnaire were distributed to 120 voluntary participants. Among them, we had two groups. We are divided into two groups. 62 were the patient group and 58 were the donors group. The questionnaire consists of the 30 closed-ended questions with the options based on the Likert scale, where 80 is considered as a neutral, and more than 80 was considered as positive, less than 80 was considered as a negative. Coming to the results part, when handling this residual blood samples, which have been used for the research, the donors or the patients, they agreed, like almost like 64%, they agreed the research can be conducted. And also, if when it was questioned about the beneficial to the society, that altruistic behavior, they were 74% of the people agreed for the research on their blood samples. Regarding the broad consent, which is a major ethical issue when it comes for this handling these samples. So when questioned about the consent taken at the time of drawing the blood for the investigations of the blood donation, is it okay, the adequate to take up the blood samples, the residual blood samples for the research? The 68.66% of them agreed then it is okay to do the research on their blood. And result of any kind of the study which is carried on their blood, they agreed that 80% agreed that that should be informed to them. And any almost like 90% of the people agreed that any results of genetic study should be informed to them. Coming to the ownership and the decision making, you can here there is a clear disparity shown here. The some subjects, like some subjects, most of the subjects, like 44.16 of subjects, thought that the laboratory can be the owner of the residual blood samples. However, when the question was asked regarding the fate of the residual blood samples, the patient group or the blood group, they agreed it can be the they wanted to be the owner of the blood samples, these residual samples. However, there is still a confusion part on this, because they were not aware, oh, this can also be done, the residual samples can also be used for the research purpose. So coming to the participants opinion about the storage and sharing the residual blood samples. So if the question was asked whether the blood samples were stored for 10 to 15 years in the biobank, 54.99 percentage agreed. And if the question was asked whether this blood samples can be shared within the India, to the researchers to conduct the research purpose, 56.66 agreed. And when it was asked about outside India, like sharing these samples outside India, researchers 60.82 percentage disagreed. About the marketing, coming to the marketing, the residual and the stored blood samples, the marketing. So most of them, they didn't agree for the marketing of their blood samples. And if it was asked if it is for the benefit of the society, then they were like, okay, they gave the consent. So you agree if the researcher uses the blood samples to develop a diagnostic kit with a huge marketing potential and benefit. So that some 40 percentage people agreed on that. And so this is the questionnaires which asked about this, different types of tests or the different type of research on their blood samples. So you can observe that mainly related to the cancer therapy or the gene therapy and Alzheimer's disease, they have a maximum percentage shown here, showing that they are interested to know about the research carried out in these fields. Coming to the actual results of the attitude scores between the donors and the patient, the blood donors had a positive attitude. Like actually, both had the positive attitude towards the research on their this one, but it was more on their patients who gave blood for the investigations. They were more positive towards the research purpose. Both male, coming to the gender first, there is both males and females, they had a positive attitude for medical research on their residual blood discussion. So the participants showed positive attitude towards their residual and the stored blood samples for scientific purpose and research. And this was but same results were seen in some of the studies, previous studies, like Annan et al. Genetic research from Gaza Strip, Evans et al. And all. Their motivation behind is mainly the altruistic behavior, like it is beneficial to the society. If any medications are developed for this genetic disease through their research, that can be helpful to the society. So maybe this is the reason why they were agreeing for the research on their samples. More than 68% participants agreed that consent taken at the time of drawing blood is adequate to carry out the research purpose. Coming to the discussion part, the end of the conclude part, the ownership and decision making about the secondary use of the residual blood samples were mixed. Accordance in justifying this in accordance with the Universal Declaration of Human Rights, it is recognized that a person, laboratory, or institution cannot own another person's body or the body parts. 44% disagreed to consider the laboratory as the owner of the leftover samples. The attitude scores of the patients were more compared to the donors for the research on their blood samples, and this was in accordance with same the Adan et al. And the Amanzima et al. On cancer patients. Concluding, so these stored blood samples along with data are the increasing being used for the genetic research. Newer concept of ethical issues like genetic privacy, genetic confidentiality, and genetic exploitation have raised complexities to compare to the other type of research. So findings of genetic research not only involves the individual from the samples whom where it is obtained, but also includes this entire family or the racial community he comes from. My studies concludes that in India, along with all the ethical issues involved, the additional problem with the low level of health literacy among the population, where we can clearly see that who was the owner of the blood sample, or whether it can be given for the beneficial of the society. When many questions were asked, so they were not sure about that. Maybe after henceforth, when they are giving blood for that investigation, or for as a donor, if they will be having this second thought henceforth. The level of literacy among the population and inability of the general public to distinguish between the treatment and the clinical research, and also the general public are not aware of the research on the biological material. So this was my topic, and these are my references. Thank you, everyone. Thank you, respected dignitaries, for allowing me to present my topic. Thank you. Am I audible? Hello.